Hey, I'm Mike Granda. I'm one of the clinician researchers here at University of Washington and Kidney Research Institute. And I'm going to talk about the gut-liver-kidney axis of uremic solutes. So I have nothing to disclose. I'm supported by grants from the American Society of Nephrology and the University of Michigan. So an outline of what I'm going to talk about. First, I'll explain the background of uremic solutes and then talk about the individual components of the gut-liver-kidney axis and how they each play a role in the generation, transformation, and ultimately clearance of those solutes. I'll talk about how they relate, including some preliminary data from a study that we conducted, and then just a quick overview of some of the therapeutic strategies that I'll have covered. And so there are multiple definitions for uremic solutes. This comes from the European Uremic Toxin Work Group. And the uremic syndrome is attributed to the progressive retention of a large number of compounds, which under normal conditions are excreted by the healthy kidneys. These compounds are called uremic retention solutes or uremic toxins when they interact negatively with biologic functions. Many researchers and work groups have sought to standardize the definition of what determines a uremic solute. In general, to be labeled a uremic toxin, it has to be identifiable chemically. It has to be measurable in human blood. The level has to increase in accordance with progressing kidney dysfunction. There should be a relationship between the level of the toxin and the development of uremic symptoms or manifestations. And there should be a plausible mechanism by which the solute can cause biologic injury. And again, while there are different ways to categorize uremic solutes, it's common to label them by their physical and chemical properties. The first category are the generally small and very soluble solutes, which include things like urea and creatinine. These solutes are very easily removed through hemodialysis and are cleared well by filtration, although some tubular secretion might occur. The medium compounds are larger, and these are typically compounds related to proteins or immunoglobulins. The prototypic example is beta-2 microglobulin. These are cleared to a lesser extent through hemodialysis, and they tend to accumulate with dialysis vintage, which can lead to progressive manifestations like amyloidosis, for instance. And then the last category include many of the most well-known uremic compounds, which are the highly protein-bound uremic toxins. These tend not to be very water soluble, and they need carrier proteins like albumin to be delivered to the proximal tubules. Dialysis poorly clears this third category, as very little of those compounds exist freely dissolved in the serum. And so uremic toxins have been linked to dysfunction in basically every organ system. Experimentally, it's been shown that high levels of uremic toxins can worsen kidney disease itself. A lot of the increased risk for cardiovascular disease and CKD is mediated through uremic toxins. Uremic toxins exert metabolic pressure by reducing insulin sensitivity, and part of the uremic milieu includes dysregulated hormones like parathyroid hormone, which can lead to bone disease. And so overall, a lot of the symptoms and complications of CKD have been associated with uremic solute levels, which ultimately lead to higher mortality and worsened quality of life. Many of the putative uremic toxins depend on the gut-liver-kidney axis for generation and clearance. In no particular scientific order, I chose four well-known uremic solutes, all of which in some way depend on amino acid metabolism by bacteria that's contained in the gut and have downstream pathologies that are known to occur in CKD. All four of these solutes have known in vitro effects, but they've also been associated with worse symptoms in CKD patients an increased risk of death. And so moving on, I'll talk about each component of the gut-liver-kidney axis and how they interact to determine the plasma levels of these solutes. So first up is the gut, which harbors a very diverse colony of different types of bacteria, which we call the microbiome. The microbiome is a symbiotic ecosystem that protects against invasion from pathogenic organisms. And it helps to digest certain food products and facilitates the absorption of amino acids and micronutrients. The composition of the microbiome is highly variable between individuals, and it's known to change in accordance with factors like diet, medications, the environment, and the presence of certain comorbidities. Most of the uremic toxins that I'm covering are byproducts of the amino acid metabolism of this gut bacteria, which lead to intermediates, which can themselves be toxic and generally require further processing before they can be cleared. So taking a few examples, indoxyl sulfate and indoxyl glucuronide are well-known uremic toxins that result from gut bacterial breakdown of tryptophan. The bacterial tryptophanase leads to the production of indole, which is then absorbed and converted to indoxyl sulfate and glucuronide, can't say that word, to eventually be cleared. A similar process happens with the breakdown of tyrosine, which leads to the intermediate of p-cresol. In both cases, the gut bacteria can further influence the retention and the proportion of these uremic solutes by, for instance, breaking glucuronidated solutes back down into their intermediates, which can then generate things like indoxyl sulfate and p-cresol sulfate and other uremic toxins. Besides ingested products, the gut bacteria also play a role in the metabolism of endogenous products. One notable example is the breakdown of urea, which leads to the generation of ammonia, which is readily absorbed from the GI tract into the bloodstream. And as we know, it's a major source of mortality in our liver disease patients. So as I said before, comorbidities can exert a selective pressure on the microbiome. This is known as early as the 1970s, in which patients with ESRD were recognized to have different gut bacterial populations from colonoscopy samples. In some instances was exploited to treat uremia with antibiotics. This more recent study examined gut flora by phylogenetic microarray in 12 healthy controls and 24 ESRD patients, and specifically identified the altered bacterial species that was present in fecal samples. In a separate paper, the same group matched the expanded bacterial populations to their enzymatic properties, and found that many of the bacteria in higher abundance in ESRD patients were those that could specifically break down the higher levels of urea, uric acid, and they also showed a higher preponderance of enzymes that generate uremic toxins. So interestingly, while CKD can select for certain bacterial populations, the actual importance on the levels in blood are unclear. The gut flora populations haven't been conclusively linked to the higher levels seen in CKD patients. In particular, in this 2020 study, they did note that plasma levels increased across severity of CKD. However, interestingly, the urine concentration did not, in accordance with the reduced clearance, but neither did the fecal levels of amino acids or uremic toxins. And so they postulated that only really the reduced kidney clearance was responsible for the high circulating levels of these uremic toxins. While the source of all uremic toxins isn't necessarily clear, we know that the gut flora plays a significant role for many solutes, and all of the solutes that I'm talking about. This study was particularly drastic in showing the role the gut flora plays. It measured uremic solutes in healthy and ESRD controls, as well as ESRD patients after colectomy. So far, this study shows the most dramatic reduction of uremic solutes, in some cases below the levels seen in healthy patients. And this is assumed due to the reducing of the gut flora population. So many of the investigated strategies that I'll talk about rely on reducing the population or the activity of the gut flora in generating uremic toxins. Looking at dietary changes, there is good evidence that low protein diets reduce the level of uremic toxins. In the study, patients were on a restricted diet of 0.6 grams of protein per kilogram of body weight per day. The patients in the study were separated into groups by their adherence to the diet, which they monitored by 24-hour urine urea nitrogen as a marker for their protein ingestion. And it found a significant reduction in at least 2 amino acid-derived uremic solutes. Looking further into the role of the diet, this study set out originally to determine if using a specific dialytic maneuver, hemodiafiltration, led to improved clearance of protein-bound uremic toxins. To that end, it actually showed no effect, but observationally, it captured a large number of patients that were on a vegetarian diet. And so they report these results showing that those on a vegetarian diet did have significantly lower levels of indoxyl sulfate and p-cresol sulfate. That being said, there are specific limitations of the study. That the power was somewhat limited, and the effect might have just been due to lower protein intake overall, which wasn't specifically measured in this study. Quickly, in this study, patients received 6 weeks of fiber supplements, given its known role in limiting the absorption of harmful compounds. The effect they saw was pretty small and was generally limited to indoxyl sulfates. So moving on to the next step of the process, the liver's first pass metabolism plays an important role in limiting the toxicity of ingested substances, and it transforms them through different enzymatic steps. Several of the solutes are rendered more soluble or more easily distributed to the kidneys for elimination by the liver. And lastly, the liver synthesizes albumin, which I said is the main carrier for many of the uremic toxins. So continuing with the examples that I used before, p-cresol results from tyrosine metabolism by gut bacteria, which is absorbed into the portal circulation. In its sulfated form, p-cresol sulfate is then transported to the kidneys for elimination. Indole, on the other hand, from tryptophan metabolism, can undergo transformation through different enzymatic steps, including the CYP450 enzymes that we know are relevant to drug metabolism. Ammonia, which is predominantly from gut bacteria metabolism, is converted in the liver, and to a lesser extent in muscle cells and other organ systems into urea, which we know is cleared very efficiently through the kidneys. So given its role in transforming gut flora waste products, it's plausible to assume the conversion of intermediates to uremic toxins might be altered in liver disease. And so this study by Lin that I'll mention a few times looked at plasma levels of indoxyl sulfate and p-cresol sulfates in 115 patients that had both kidney and liver disease of varying severity. I highlighted this excerpt from their paper in the patients that had no appreciable kidney disease. And in this subset, they found significant decreases actually in the levels of indoxyl sulfate and p-cresol sulfate with worsening liver function. So given the fact that kidney and liver disease coexist in many patients, it's interesting to think about the role of albumin along the gut-liver kidney axis. Despite being so ubiquitous, there is some evidence that uremic toxins can saturate albumin in ESRD patients. There was a 2009 study published in CJASN in which uremic serum was actually spiked with additional purified indoxyl sulfate or p-cresol sulfate, and found that even with small additions of those compounds, it could easily displace toxins that were bound to albumin in the uremic serum. In the LIN study, the level itself of albumin didn't have any significant effect on the free or total levels of the two measured solutes, indoxyl sulfate and p-cresol sulfate. Besides plasma levels, even less is known about how albumin levels in the plasma or urine might alter the actual clearance of these solutes. Like uremic toxins, a lot of common drugs require protein binding to be distributed in the body. We know from crystallography studies that the specific binding sites for uremic toxins are shared by many drugs. Yeah, I found that among the binding sites, many overlap between uremic toxins and common drugs. In human patients, I won't delve too far into it, but plasma levels of uremic toxins have been shown to alter the metabolism and renal and non-renal clearance of several drugs like furosemide, tacrolimus, aripiprazole, mycophenolates, and several others. So obviously the most important stop on this axis, the kidneys are the main route of elimination for all uremic toxins. While many of them can also be filtered, the majority rely on tubular secretion for clearance. When the kidneys fail to clear these solutes, the increased levels of uremic toxins select for toxin-generating bacteria in the gut and can alter drug and toxin metabolism in the liver. The interplay between the gut and kidney is also important. The CKD is thought to increase the absorption of uremic toxins and potentially increase the risk for bacterial translocation by inducing intestinal permeability. And this is known as the leaky gut effect. Many of the uremic toxins are cleared by the kidneys at remarkably fast rates that far exceed glomerular filtration rates. In general, the solutes with the most rapid clearance have only partial protein binding, and so they can be both filtered and secreted simultaneously. Solutes with significant protein binding, on the other hand, can't be cleared by dialysis, and so these tend to accumulate over time. So if we're putting all of these steps together, the gut microbiome, which dictates the type and amount of uremic toxins generated, is altered by numerous patient factors like CKD and liver disease. Liver disease limits the ability to metabolize or solubilize toxin intermediates, which can itself cause kidney disease. Uremic solute clearance is limited in kidney disease. Only transplant restores secretory clearance, as dialysis only recapitulates filtration. We know that CKD is a modifier of the microbiome and may enhance toxin absorption and affects liver enzymatic activity. So has a lot of wide-ranging downstream effects. We know that high uremic solute levels are associated with CKD progression. And finally, many of these processes occur simultaneously, but the existing studies tend to focus on only a single aspect of the gut-liver-kidney axis. So it's interesting to note how markedly the levels of uremic solutes change in accordance with both kidney and liver disease, not being classically associated with liver disease. So going back to this LIN study, as I showed earlier, the levels of indoxyl sulfate and p-cresol sulfate tend to drop dramatically with worsening liver disease. But across the stages of kidney disease, we see there's a significant increase in the levels of indoxyl sulfate and precel sulfate, really only in the earliest stages of cirrhosis. In the last stage of cirrhosis, the Child-Pugh category C patients, the levels do increase as kidney clearance gets worse, but the absolute level of plasma indoxyl sulfate and precresol sulfate are actually extremely low, and they're below that of the least sick counterparts. So exactly how this happens is slightly unclear, as I'll talk about next. So I'll talk about some data that we have from our Icicle study. This was designed to address the question of proximal tubular secretory clearance in liver failure. Our group started the Icicle study, which enrolled patients from 2019 to 2021. Participants were recruited as outpatients across varying degrees of kidney and liver failure. This resulted in 39 participants total, about 2/3 of which were men in their 50s and 60s. To avoid having a predictor that directly accounts for the outcome, we used the Child-Pugh score instead of MELD, which is more commonly used clinically, because MELD itself includes a term for creatinine, whereas Child-Pugh does not. We matched the participants to non-liver disease controls in our study's sugar and proclaim by eGFR plus or minus 10. And then we measured urine and blood levels of several highly secreted solutes, most of which are putative uremic toxins, to estimate their secretory clearance. So first looking at the plasma levels, we saw a drastic difference in plasma levels of many uremic solutes when we compare all Icicle patients to all controls. Mimicking the LIN study, both indoxyl sulfate and precresol sulfate were drastically reduced in the liver patients. Interestingly, two solutes that failed to reach significance, dimethyl uric acid and xanthosine, are diet-derived from purine metabolism, but aren't known to have significant contributions from gut flora, whereas many of the other solutes do. And the association of the plasma levels didn't change significantly when adjusted for different factors, including the degree of kidney impairment by eGFR, by age or sex. While we showed a similar trend of plasma levels of uremic solutes decreasing, as in the LIN study, across patients with cirrhosis, in our subgroup, in our group, the differences didn't reach statistical significance, although they did tend to decrease with advancing cirrhosis. This remained true even when adjusting for potential confounders like eGFR, albumin, age, and sex. And so when we look at the resulting clearances, we see something that was very unexpected. While the plasma levels of the solutes decreased in patients with cirrhosis, the clearances in several instances actually tended to increase in patients that had both liver and kidney disease, which is definitely unexpected. The reasons for this are unclear. It doesn't seem very plausible that secretory clearance is actually increased in the liver failure patients. Generally, secretion and filtration change together, and in our model, even after adjusting for GFR, the association is basically unchanged or barely attenuated. One thought was that the plasma levels are just so low that they've artificially increased the calculated clearance. It might also be possible that the low levels of multiple solutes has led to less competition for secretory transporters. Along that line, many of these solutes require the liver for transformation. And so, it does make us wonder if there's a higher concentration of circulating intermediates that failed to be transformed by the liver which aren't being measured in this study. The role and the clearance of those intermediates isn't obvious from this data and weren't measured, but could be addressed in the future with screening methods like metabolomics. Going back to another phenomenon I briefly discussed, many of the liver patients are on antibiotics, primarily for SBP prophylaxis. So as we talked about, antibiotics can exert a selective pressure on the gut flora and have been investigated in the past as tools to reduce uremic toxins in CKD patients. I looked at the effect of antibiotics on plasma levels across Icicle patients that were on ciprofloxacin or rifaximin, and did find a tendency for some solutes to be lower in antibiotic patients, but none of them actually reached significance. This is again in keeping with the general finding that all of our cirrhosis patients had just extremely low uremic toxin levels. So going back to the way that all of these pieces sort of fit together, I've talked about a lot of different investigated strategies along the axis to treat the presence of the uremic toxins. Starting with the microbiome, we know that diet can influence the populations of bacteria in people's gut flora. The use of pre and probiotics has been investigated and showed very limited effects, and antibiotics have also not really demonstrated a consistent effect. Limiting the production of those toxins is one sort of strategy that is in common use by protein restricting CKD and ESRD patients. And giving fiber supplements has also been investigated and showed a very minimal effect. There are binders that limit absorption, such as AST120, an activated carbon supplement, as well as amylose starches, and neither are in clinical use. In terms of the transformation of the solutes, really the main strategy is to limit promoters of liver disease. And in particular, there's an association between CKD and non-alcoholic fatty liver disease. And so monitoring patients and limiting the progression of liver disease might be another strategy. And then finally, for clearance, obviously limiting CKD progression is important to us. Transplant restores the clearance of many of these uremic toxins. And then there are a lot of dialytic maneuvers that have been investigated, including some that are under investigation here to help clear these protein-bound uremic toxins, none of which are in active use. So in conclusion, the gut flora seems to play an outsized role in uremic toxin generation. The reduced liver function tends to decrease the level of uremic toxins significantly, even in CKD, although the biologic effect is unclear and presumably leads to higher levels of intermediates instead, which are less studied. Our findings confirm exceptionally low levels of uremic toxins in liver patients, which seems to be minimally affected by GFR or the degree of kidney impairment. And then finally, our findings of proximal tubular secretory clearance in liver patients is surprising. And so more studies are warranted to see how that changes in accordance with liver disease and any symptomatic or downstream effects of changes in secretory clearance. So thank you all for letting me speak. And I want to make sure to acknowledge Brian Kestenbaum, who's my primary mentor, and several other people that have played very important roles in this project.
Thank you, Dr. Granda, and clearly really promising research direction. There's a lot to do. Questions for Dr. Granda? Well, I'm personally curious about what you see coming down the pike in terms of dietary recommendations. The gut microbiome is on our minds, and I think a lot of patients, are also in tune with this. Do you see, what do you see as promising for the near future? And who would we think about, most helped by these kind of dietary interventions or other ways to cultivate the gut microbiome?
That's a good question. I think, a lot of things haven't shown a particularly large effect, aside from sort of limiting the precursors of the waste products, so low protein diets. The effect of medications is also under investigation. So things that we're commonly giving to CKD and ESRD patients might play a role. So things like oral iron might promote different bacterial species in the gut that might change production. Patients that are on phosphate binders, things like that, might also change the population. So I think that's one area that hasn't been fully fleshed out, is sort of the medications that people are exposed to, besides the known role of antibiotics. Leah said, how about fecal transplants? I've seen only one study. There might be more. I can't say that I did an exhaustive search for that. And I don't think that there was a significant effect from fecal transplants, I think patients were followed for a relatively short duration. So I think it's hard to say. There might be more data that I'm not aware of, but I haven't seen a large role for fecal transplant.
Mike, following up on Kate's question about diet, most work, including your and I's work, is focused on proteins, of course. And if you restrict dietary protein, if you keep calories the same, you have to have more carbohydrate or lipid. Is there any role for gut microbiota in lipid and carbohydrate modification in terms of generating toxins that might be relevant?
I'm not sure that I know enough to tell you that. A lot of the solutes that I talked about are related to amino acid metabolism. Some of the uremic toxins that we're aware of are sort of secondarily related to free fatty acid metabolism. So I think things like isovalerial-glycine, if I'm remembering correctly, whether or not that's performed by the gut flora, I guess I'm not sure. So I think it's hard to say, but that is an interesting question. I'll have to look into that.
Well, I'll move on for time. There's one more or a few more questions in the chat that you might be able to address briefly, Mike. But we'll move on to our next speaker.